Two researchers from Boston's Children's Hospital, known as Dr. Leonard Zahn and Charles K. Kaufman, discovered new findings regarding how cancers may be developed. Dr. Zahn and Kaufman discovered a single cell in zebrafish that would later turn into melanoma. According to the Mayo Clinic staff, melanoma is the world's deadliest form of skin cancer. It develops in the cell's melanocytes that produce melatonin, which is the pigment that gives your skin its color. The first signs or symptoms for having melanoma are often a change in an existing mole or the development of a new mole. However, melanoma doesn't always begin as a mole. In cancer biology, a tumor begins with a group of precancerous cells that share genetic mutations. Cancer growth occurs when unrepaired DNA damage and skin cells trigger mutations. These mutations lead skin cells to multiply rapidly, which then forms malignant tumors. As you can see from this photo, the cancer cells are dividing much more rapidly than the normal cell division. How exactly can melanoma be detected? Melanoma is detected by the ABCDE method. A is for asymmetrical shape of the mole. B is for irregular border, how uneven the mole might be. C is for changes in color. Look for growths that may have many colors or uneven distribution of color. D is for diameter. Lastly, E is for evolving, which means looking for changes over time, such as a mole that grows in size or that changes color or shape. Moles may also evolve to develop new signs and symptoms such as itchiness or bleeding early. Although the cell was not cancerous yet, it was still surprising. Dr. Zahn and Dr. Kaufman realized that the cell reverted to an embryonic form when it could have been developed into any other cell type. As it began to divide, cancer genes took over and the single cell turned into a massive tumor. But what besides powerful cancer genes is needed to make cancer? Dr. Zahn and his colleagues began to investigate melanoma by creating it in the zebrafish. They used zebrafish because they were tiny, transparent creatures that allowed the researchers to see cells or organs without cutting the fish open. The researchers would put human BRAF and P53 genes in every melanocyte, the pigmented skin cells of the fish. If those genes were all that was needed for cancer, the fish would explode with melanoma. More was needed than just the BRAF and P53 genes. Researchers only found 1 to 3 melanomas per fish. The cells that became cancerous had turned on the gene crestin. Crestin is normally activated only in cells that are part of the neural crest. Typically, crestin production ceases once the cell becomes mature. With mature cells still producing crestin, the cells were malleable, and what they would eventually become. Crestin was probably never shut off as a result of mutations in the cell. The mutations could have occurred because of an error in the cell cycle. The cell cycle starts at G1. At G1, the cell checks for cell size, proper nutrients, growth factors, and damage to the DNA. The cell then proceeds to the S phase. After the S phase, the G2 phase starts. At the G2 phase, the cell checks for damaged DNA and also to make sure that DNA is complete. The cell then proceeds to mitosis. Apoptosis happens by breaking the nucleus into many small parts. Organelles are also broken down into many small parts also. These small parts are eaten by phagocytic immune cells. When functioning properly, apoptosis would remove potentially cancerous cells. This did not happen, and cancerous cells started to develop in the zebrafish.